minimum, and it might go much deeper than that. And so this is the classification scheme that we have in the guidelines. It's by the electronics package and how it relates to the retention elements, and Mike already went over that. I'll focus most of my talk about these type A tags, but just equally important, do we have some kind of a pointer, or can I use my finger?
We can see.
This,
Yeah, you want to see the error?
Yep.
Yeah.
you can see it online too? Okay, so down in the bottom hand side, you can see a killer whale, and it's got a cavity through the sagittal section there. You can see this cross section. Can't make my mouse move. Cross section here, and then that's blown up in this upper part here. So you can see the dorsal fins up on the top. There's a spinal vertebral process in the middle. And then you see the muscle around the spine, and then either the subdermal sheath, which is how technical people call it. It's also often called the fascia sheath that surrounds the inner organs of all cetaceans, especially well-developed interdonticetes, but mysticetes have it as well. And then you have the integument, the dermis, and the hypodermis. So here I have a bunch of type A tags illustrated, but you can see by classification scheme in their anatomical position, they're not all the same. So here we have a tag, type A tag, that has a very short retention barb that matches about the blubber thickness of the animal, so that the very tip or the most distal part of that attachment element does not reach the subdermal sheath or the fascia. So that we would call a suprafascial tag. All of the tag is above the fascia. But here's another type A tag with a much larger retention element. This tag has been used with a lot of animals by Mads Peter Heidi Jurgensen, and very successfully on bowhead whales, for example. They've checked animals for almost 2 years with tags like this. And that tag has its retention element in this case that is much longer than the average blubber thickness. And so it reaches below the subdermal sheath or the fascia, and that type A tag is a subfascial tag in this case. And so a type A tag could be just in the blubber, a suprafascial tag, or it could be a subfascial tag. Now, most type C tags, the consolidated tags, are designed specifically to be subfascial tags, and most of the type A tags have been designed specifically to be suprafascial, but that's not always the case. With type C tags, you could have a subfascial tag. So for example, on the left-hand side here, so this time we have a humpback whale with much thicker blubber, say it has 15 centimeters of blubber. If you have a type C tag with 2 AA batteries, it'd be about 29 centimeters long in this case. So half of the tag is below the fascia. But you could have a type C tag with just a single AA battery. In this case, all of the tag would be in the dermis and hypodermis, so it would be a suprafascial tag. So just, it's important to kind of keep this in mind when we're talking about these different tag types. From the whale's perspective, it doesn't give a damn. It's what's really in the whale's body and where it is, and how that affects its impacts on its health will probably depend a lot more on anatomical location, assuming you've read the best practices and you've sterilized it, you've used good materials and practices of delivering the tags. So quickly, I'll just talk about tag delivery methods. Of course, in capture animals, that's a much better way to do it. Pretty hard to capture a North Atlantic right whale. So obviously, we're going to talk about remote methods for that. The standard ones that you've already heard people talk about, poles with limpet tags. We also use crossbows or pneumatic projectors of various types. All of those work really well. There's experimental techniques. Somebody in the room has tried this. Another group has also tried it, but I don't think anybody's ever succeeded to lasso the tail of a whale and get a tag on it. But there are other experimental techniques that might be important to think about when it comes to North Atlantic right whales. In the mid-'80s, Bruce Mate hired a radio-controlled helicopter pilot to try to place a type A tag on humpbacks. That didn't work out so well, and they tagged a wooden boat instead of tagging the whale. But you've now heard about using Drones to drop suction cup tags. You can use a drone to drop any kind of tag. So given the difficulties with approaching specific right whales, and that might be an important thing when we say, you know, it can only be e.g. 4092 and 4054 or whatever, or only males, chasing around right whales in a boat all by itself is a problem. And if it were me, I think I'd probably rather have a little sliver stuck in me than this big, huge boat chasing me. And sometimes it really does involve approaches that look like chasing, even though I think most of us who are in the boats would never want it to be called chasing. So how we deliver the tags is a pretty critical thing. I think some other folks have covered a few of the things about the history. This is the tag that Bruce tried to drop from a helicopter. It involves 12 sutures that yank in there, but it's a Type A tag. This was a later version of his tag that I think they managed to tag a right whale for about 22 days with that. Eventually, they evolved to this one in the middle here, another Type A tag where all the electronics in there in that cylinder. I think they might have gotten almost 40 days on a right whale out of that. Don Kroll had a tag that's also a Type A tag shown here on the right, where there's an anchor that I'm pointing to right now, and then a tether or a loose attachment to the electronics. And that potentially can have some advantages as well. None of these tags, other than bottom left and that right one, lasted more than, a few days. There's lots of other Type A tags. More recently, Mark Baumgartner developed this short-term dermal attachment tag. I think they got up to 5 days on a humpback once. And then off-the-shelf pop-up archival satellite tags that are more typically used on fish have been applied to Pacific whiteside dolphins and pygmy blue whales in Australia. And in both those cases, tags didn't last for more than a few days. So that brings me to talk about the limpet tag, which is a Type A tag that is an acronym for something that doesn't properly describe what it is. It probably is more accurately, if you're going to use environment technology, a barnacle style tag, like all Type A tags are. But limpet sounds much more innocuous, so I like it. It also stands for low impact, minimally percutaneous external electronics transmitter. Originally, we developed it for tracking killer whales that were feeding on stellar sea lions. And the first successful deployments of what now looks like the off-the-shelf limpet tag was done in Antarctica. We designed it to go, designed it to be embedded in the dorsal fin, and we can use air guns, as you can see on the left, or crossbows or poles. Over the years, we've gone from kind of handmade cobbled electronics to working with wildlife computers to have everything off the shelf, including location-only versions of the limpet tag and ones with dive depth recording and transmission, and finally GPS. And the robustness and the design of the tags has developed over time, thanks to this ONR not funded study, one of which that Mike had mentioned, which was a collaboration between myself and Greg Shore of Mari Patel when he was with Cascadia, and Robin Baird, and then also Brad Hansen at the Northwest Fisheries Science Center, and then a couple of bioengineers, Rajat Mittal and Lawrence Halls. We had four main goals with that evolved our understanding of how the limpet anchors react with tissue. So we did a lot of work on how different kinds of tissues, blubber and dorsal fins from different species affected the ability of those retention elements to actually do their job. Then the bioengineers helped us both with computer fluid dynamics as well as physical models to understand how best we could allow the external forces to minimally impact the retention of the tag. And then we looked with real tissue as well as modeling to look at the in situ performance of those elements on the tissues in which they're embedded. And finally, perhaps the most important that I'll spend more time talking about tomorrow, the follow-up studies to look at the tagged whales, to look at wound healing and the effects of tagging. I won't have time today to talk about the thermal imaging that we did with healing, or really to talk about the survival and reproduction studies, other than to say, with survival and reproduction, we specifically looked with Rob and Baird at the whales that we tagged in Hawaii, specifically short fin pilot whales and false killer whales. At the time, I think we had well over 100 short fin pilot whales and about 30 false killer whales tagged with limpet tags, with average durations around about a month to 45 days. It turns out with the help of Alex, we did some simulation studies, and it turns out that our power to detect a difference in survival or reproduction is vanishingly small. We would have to tag a really large proportion of the population, wait many, many years because these animals don't reproduce very regularly, and have a huge treatment effect. For us to be able to detect a significant difference with animals that have adult survival around 95%, we'd have to tag way more animals than we tagged, and the treatment effect would have to be at least 17 to 25%. If it was even 5%, we'd have quit tagging already. So, you know, we know it doesn't look dramatic, but it would have to be dramatic for us to statistically prove it to you. So that's a tough one to keep in mind when you're doing this stuff. And maybe Alex has more up-to-date information on this kind of thing, but it's hard. Some of the other stuff that we did, how am I doing on time? 7 minutes. Okay. Looking to try to better understand what's happening with tags and why sometimes they last an hour and sometimes they last a year. We started out with this pretty simple model where over time we expected the hydrodynamic pulling force to increase because the tag at first is really well adhered close in the boundary layer on the whale, but it slowly pulls out over time as the tissue necroses around the darts. So it's up in the water column, it's getting hit by more water that's moving faster. So over time, that hydrodynamic pulling force should go up. The barb retention force we expect to go down a lot because in the very beginning, those barbs that stick back are well adhered into good, well joining tissue, but that tissue was necrosing. We just killed those cells and now we got salt water and all the bacteria and viruses coming in there. So we expect the barb retention force to go down, but we haven't properly characterized it. Mathematical simulations and actual water flow studies, we measured all of those things. We measured how hard the tags are to pull up when the tissue is fresh and old. We smack into it with. It turns out that in the cases where the tag stays on for like a year, eventually the tag, in this case, you can see a short fin pilot whale with the tag just barely hanging on to the dorsal fin. Can everybody see that? Okay. In that case, the tag stayed on a long time. That particular one, I don't remember, like maybe 6 months. So in that case, it is possible that eventually the actual retention force is matching the hydrodynamic pulling force in the water actually pulls that tag out. But in the vast majority of time, probably 99% of the time, the barb retention force is actually way higher than the forces that are acting on the tag other than when it bangs into something. So when it's swimming through the water or jumping even, those pulling forces are way less than the force that the tag is holding on. And in fact, what really happens is hydrodynamically, it's kind of complicated because when the tag is really well attached, water is actually flowing over and there's a lot of lift. When it first lifts up a little, water can go underneath it. So it actually is pulled less when it's pulled out of the whale a little bit. But in fact, eventually there's some sudden impact force and the pulling force is actually banging into another whale usually, and then the tag falls off. And that's really well reflected. This just a quick picture. We've tagged, I think, at least 32 species now. All the green check marks are the species that have been tagged. Over on the right-hand side are, can't remember how many species are up there, 7 or 8 species that have a sample size of at least 10 animals that had limpet tags with the darts that are 7 centimeters. I should have said we also have that you saw on there. But for all of these species, they had 7 centimeter darts tagged either in the lower part of the dorsal fin or the dorsal fin. And you can see that each color codes a different whale. And you can see there's a lot of variation, both within species, between species. Some species get rid of their tags always really early. Others have it long. This actually was data that we compiled in, I think, 2016. We now have a few pilot whales that have carried their tags for almost a year, one just a couple of days short and one a few days more than that. But lots of them have carried at short fin pilot whales. Back up 1 minute later, the tag is gone. So it can last 1 minute or it can last a year. Most of the time it's on the shorter end, but the average is actually about 40 days. For baleen whales, here's one example where we have a big sample size, 82 fin whales that we've tagged, and this is not including the fin whales that Veronique's group have tagged that I've been collaborating on. The minimum for those fin whales is 1 day. The maximum is 230 days, but the average is about 35 days. And you can see the standard deviation is huge. I'm trying to understand that as a critical part. But overall, they actually do pretty darn well, and rarely do we see any complications. But that's the nice side of the story. At least one whale that was previously tagged died shortly after its tag fell out. And an expert panel of veterinarians concluded that the most likely reason for that whale dying was that a fungal infection was introduced by the tag. Now, unfortunately, this is something that all invasive techniques that are remotely applied to any cetacean would risk, because anytime we're remotely doing things, at least with our current technology, we're not preparing the surface. We know for a fact that that fungus that killed L95, southern resident killer whale, is found sometimes on the surface of whales or in the water. So if you take something and poke it in without preparing the surface, that's at a risk of, even if it's not even on the tag, if you properly sterilize the tag. And that can happen with type A tags, type B tags, type C tags. It can happen whether they're in the blubber or not. The vets can argue about what's going to be worse for introducing microorganisms. But in any case, if we don't clean the surface, the risk of infection that we cause is always going to be there, even if we do the proper sterilization. And in the vast majority of previous tagging, sterilization wasn't done at all. I can say that even when I first started it, my vets told me that I didn't have to sterilize the tags. I just had to disinfect them. And there's a big difference. You should read the paper if you don't understand it. This is another type A tag. I'll just finish up. So this basically is a D tag that can last for up to 2 weeks. So all the arguments about how wonderful suction cup tags are, and they can only do, well, this tag that Mark Johnson and I developed with Wildlife Computers can record sound. It only has a single channel. Can record sound, and it has been applied to short fin pilot whales, cuviers, beaked whales, and fin whales so far. On average, with short fin pilot whales, we've done about 11 deployments, and on average, they stay on for about 6 or 7 days. The longest has been 21 days. Usually, we try to drop it much shorter than that. So finally, North Atlantic right whales. Clay already covered this. I've been collaborating with them in 2015 and 16 down in the Southeast US. We had one tag that we tagged with a limpet tag, just kind of off the shelf, just to see if this could work when it was only in the blubber. Lots of those other whales we did tag in the blubber, and generally we find that it's not in the dorsal fin, they don't do as well. And we did have one juvenile animal that just took off immediately, didn't touch anybody else, and traveled all the way up to Nantucket, where I think one of the previous speakers talked about, turns out to be an important feeding area. And that was 51 days. And that was the first whale we tagged. Like, yoo-hoo. But then we had one whale that lasted 2 days. And tomorrow I'll talk a little bit more about what we did in the Gulf of St. Lawrence. Key things are that we did pre-tagging health assessment. Am I out of time? Yeah. Okay. So, we prepared something and we said, this is what we're going to do in advance. And we also had a post-tagging monitoring plan that we distributed to the consortium and all the collaborators. We sent out ahead of time information on what kind of photographs we would like to get. In fact, we had very few during the time that the tags were attached because the whales were just flying, and we didn't get much in the way of tagged whale photographs. But of course, over time, with all the coverage that people have, we've got tons of. Maybe tomorrow we'll come back to talk about, these are just different types of tags, and then tomorrow we'll cover all of this. And I'll just finish. All right. Tomorrow we'll go into that But so with southeast whales, the 7 that we've tagged, all the wounds have appeared to heal well. One for sure got entangled, and probably another one. So you guys got to stop entangling these damn right whales, because I've hardly tagged any, and managed to kill some, not through tagging. But anyways, I will stop there.
Right. Thank you. Come over to the owl here so everybody can hear. All right, so we got John Kalamakitis up next. There he is. Just take a few minutes to talk about his Type A tag. Folks, again, online, if you have questions, please put those in the chat. We will have the time of this session.
Great, thanks. I'll keep us to 5 minutes. These are analogous to what Russ was talking about as the SMRT tags, but it really stemmed from our past work, working with a wide variety of tags, going back to mostly archival suction cup tags, but okay, great. But some of the needs we were facing, trying to get longer term durations with these archival tags to get the sort of rich data streams we needed. At the time, we were doing work with looking at behavioral response of animals to sound, but wanting longer term data. We were also interested in evaluating other factors, ship strike risk, as well as interaction with fishing gear. And that's what led us okay, we all agree.
Yes.
Let's go on. So that was what led us to do this modification of shifting to just taking the archival tags we were already using and adapting them for medium durations with dart attachments. And it basically consisted of adding a carbon fiber plate and then a series of darts, initially using the titanium darts that had been developed for the limpet tags. But then experimenting with those to find better ways to attachment. And we focused on 2 platforms, the Wildlife Computer TDR10 tag and the AccuSound. But then we had various modifications, adding a satellite transmitter, because with longer durations, it's a greater challenge to recover them. You have to recover them. So all of these have that. Or in the case of the AccuSound, adding a low-tech fast lock GPS, because unlike the TDR10, that wasn't part of the AccuSound, and we wanted that to be part of it as well. So we had these various add-ons. You can see it's a little bit cobbled together, and that's been our design has been to do that. We did publish a paper on our experiments with different types of darts to use and attachments, how many darts of what type. We ended up shifting to a stainless steel dart that was much more malleable and less likely to break. We had experience with the titanium darts that worked well, but since we recover the whole dart package, we could see some of the petals were breaking, as had been experienced with L95 in the limpet tag. And we were finding out with this larger tag that was not going into the dorsal fin, but onto the body of the whale, we were still getting breakage of these petals. So we ended up finding these stainless steels that were more malleable. Then I'm going to go try to go fast here. I promise to keep it to 5 minutes. The kind of stuff we were after, and just a tiny bit on applications on this. At the time with satellite transmitters, you get more positions that look like on the right. On the left, you'll see the GPS quality positions of the same animal that's on the right there. So you get just much more higher resolution with the fast lock we would get, even with a tag placed high on the body. We were only getting relatively few satellite hits a day versus getting something with every surface series of GPS quality, and look at the fine scale as well as longer movements of animals that we can get with that. That finer resolution is what let us look at kind of relationship of animals to finer scale oceanographic features. This is from a paper James Fallbush was a senior author on. The animation on the right is tracking the movement of a whale in relation to some of these fine scale oceanographic features on a finer scale than you can typically look at with a satellite tag, and showing significant association of the whale's movements to some of these small scale upwelling type events. The applications for that, for example, in terms of the shipping lanes and interactions with ships, we had two elements we were interested in. Use of the shipping lanes. This is the movements of one blue whale in relation to the shipping lanes off San Francisco over a 10-day period. This was from an AccuSound, so we were actually able to use acoustic detection of ship noise close to the whale. And the acoustics on these tags picks up a lot of flow noise, so the ship has to be really close to overcome that. And we had at least 13 of these very close near misses of this one whale just in this 10-day period of the tag deployment and use of all the different shipping lanes. And we're doing the same thing with humpbacks in a collaborative study with DFO on the border of the US and Canada in the Strait of Juan de Fuca, showing humpback whale use of shipping lanes. We also use this this kind of fine scale movement then also lends itself well to looking at reaction of whales to ships. One of the key variables in looking at ship strike risk is what degree of reaction do whales have to the approach. We had initial publication with Megan McKenna on this. We have new data on that is currently trying to get published, but is not out yet on how whales react to these close approach of ships. We did publish a few years ago a paper using, again, this tag data, because now we get lots of nighttime data, something you have a harder time getting with the short-term suction cup tags, showing not only differences in movements of the animals, that's one animal's movements, black at night, white in the day, showing wider ranging movements at night, but also very different diving behavior at night and in the day. And with blue whales, for example, showing they were twice as likely to be in the area to be struck by a ship at night than in the day. So that was an application. We've had 114 of these deployments. So a few key lessons learned. We see among the 3 primary species we've deployed them on, we do see pretty large differences in the average durations. These are durations in hours. And we've included, occasionally you have tags that barely attach and are 0, so that brings that mean down. So for blue whales, that mean of 179 hours translates to a little over a week on a whale, with durations up to 4 weeks. They're slightly shorter with fin whales and much shorter with humpback whales. The fin whales is because we use a shorter dart. Our goal, like with these other anchor tags, is to stay in the blubber layer and not go deeper than the blubber layer on these. So with fin whales, we actually use a shorter dart. Yeah, that's wrapping up. And just showing that from suction cup durations, this is across all our things and the different dart types. If you focus on the bottom panel, you'll see durations increase with both the length of the dart and the number of retaining petals. We actually got the longest retentions on the few deployments with the titanium darts, but I think that's because they were stiffer and anchored better. Okay, and so this is my concluding slide. Tag type and attachment varies depending on the primary goal, what you choose. These tags do have advantages of providing that rich data stream over longer periods of time, and they can inform those human interactions. Durations do vary by animal, and there are some challenges. We do see more animal-to-animal contact and potential damage to tags. We lose or don't recover about 10% of our tags, and that percentage was higher with humpback whales, lower with blue and fin whales. And some of that is challenging recovering tags.
Thank you. Great. All right. So we have one more talk. We've got both Alex Urbini first, and then Daniel from
Together.
Together, University of Washington. Yeah, all right. And then Daniel Plosios from Oregon State. All right. He's got his own computer hooking up here. So they're going to be talking about Type C Consolidated Tag. Reminder as they get the presentation up, if you have questions, go ahead and put this talk. We'll have a session for discussion and Q&As.
I'm going to share my screen here quickly.
Your screen, just not your presentation.
Well, okay, so hi, everybody. I'm Al Turbini. I am with the Seacoast University of Washington, affiliated to the Alaska Fisheries Science Center at NOAA, and also affiliated with Marie Cattell.
And I'm Daniel Palacios with Oregon State University.
So we're here to talk about Type C tags, and we plan on like a high level presentation today. We were asked to do and talk about what the Type C tags are, what they're, how and what species they're deployed, what can we expect from them in terms of data and duration, and also how the data provided by these tags has been used in various studies. So Mike showed this first. I think Ross went through this. This is the type that we're talking about. And this is an example of the tag. They're all of the Type Cs used today have approximately the same configuration. They differ a little bit in certain aspects. I want to highlight two important parts of the tags that we're going to discuss later. This is the electronics package. So this is a tube where all the electronics and batteries are cast, and that's the lower part of the tag is something we call an attachment system or anchor. Each one of these components have different features. Most important in the attachment system, we have retention elements and a tip. Others have talked about it, but this is what the tag looks like. These tags have been called implantable deep implant. We call Type C. Some people call them transdermal. So if you go into the literature, you're going to find different names. We're going to stick with Type C, which is what we agreed upon after the workshop we had in 2017 on tag design and development, and also in the guidelines that Mike and Russ presented. The tags are made of surgical quality stainless steel. They vary in length. The longest one of them is 11.8 inches, or about 30 centimeters. The diameter also varies between 0.78 to 0.95 inches, so that's about 2 to 2.4 centimeters. And the heaviest of these tags weighed about 390 grams. Typically, before this deployment, they're gas sterilized, and in some cases, they're coated with antibiotics. They're designed to attach to the blubber, or in most cases below the muscle blubber interface as fascia that Russ referred to. These tags remain attached for weeks to months. We're going to cover that in a little bit more. And once the tags fall off, they're not recovered. So they're not, unlike earlier with Susan, we don't recover them. And in most cases, the tag will heal and leave a small scar. I think we'll cover this in a lot of detail tomorrow. This is an example of a tag. You can see the tag right here on a southern right whale. One of the things we get a lot, I think Daniel you get, and I get, is when we talk about the length of the tag, people say, well, this tag is large. It's about 1 foot long. And if you hold the tag in your hands, it is large for a person. But if we put it in perspective with the whale, this is a 45-foot long right whale. This is the size of the tag. So for the animal, it's not that large of an instrument. And it's not very heavy either. The heaviest of the tags weighs about 0.001% of the body length of the whale, or a whale of 40 tons. So one of the things that we were asked was to talk about species that we tag and how we tag. We put together a video that is going to show you deployments in many species. Daniel, you want to comment on that? I'll comment on that as well.
Yep.
So this is what it is.
Blue whales, some of the easiest species to tag, very mellow. You can see we collect the biopsy at the same time as we deploy the tag. The red part that you saw is the carrier that sometimes stays with the tag for a little bit before it floats back to the surface. Humpbacks in Hawaii.
You can see the biopsy samples being collected. The next one back video it's a nice video of the whale underwater, and you can see the tagging, and then you can see the behavior after tagging. It's coming to the surface. And then you can see it underwater. One of the things that Russ mentioned in his presentation is there are different methods to tagging whales. In the past, we used to use poles and crossbows for this type of tag as well, but I think the community has converged in using just a pneumatic rifle.
In this case, a sperm whale, you'll see a stronger response. Sperm whales lift their head oftentimes in response to approaches, whether biopsies or close approaches. So that looks like a pretty typical response.
And these are deployments in southern right whales. This is in South Africa. The next one is a deployment in Argentina. And in the we tend to tag anterior to the or cranial to the body of the animal on right whales. It looks like the tag is going close to the head, but actually, if you look at the anatomy of right whales, the skull drops off very quickly after the blowhole. So the tags are actually going in areas where there's quite a bit of blubber and tissue there. And Daniel, I'm giving this to you now.
Thank you. So here's a brief summary of what we just saw, the places on the body and what the tags look like. I'm not sure if you can see the antenna. That's what you see in most of them. And then a little bit of the stoppers, what we call the stoppers, and they can be in different colors, but the idea is that they minimize the deep penetration of the tag beyond what we're interested in, which is basically that the tag stays flush to the surface of the animal. And of course, we want the tag to be placed as high on the body as possible, especially with humpbacks. We have found that the best place to put tags is in the dorsal hump. They stay longer. What we're going to see, this is Dave Weller's Convergent Evolution, a bit of images that we saw earlier, but this is a seminal Montgomery report from 1987. And if you look through, I highly recommend reading that report. It's very readable. And a lot of the discussions that took place in that first workshop in terms of form factor, in terms of impact to the animals, and a lot of those considerations are things that we're still dealing with. But if you'll look here on your on this side of the screen, the Jeff Goodyear design, and this was the Watkins design, which according to his own terminology was based on the discovery marks that again Dave showed us this morning. Those types of designs were implantable. These were radio tags. We were not using satellite tags at the time. I just wanted to make that distinction, but it was being discussed as the next thing to do. There were certain hurdles. One of the biggest hurdle, which is just one detail that I wanted to mention from Dave's evolution this morning, is that for using radio frequency being transmitted through the atmosphere, you need to have a very stable frequency, and because of changes in the temperature, the tags were overheating, the temperature was not stable. So there was a breakthrough when Bruce Mate worked with Telonics to be able to maintain the temperature, and that was what that punctuated moment when we were able to use satellite transmitting tags for the first time. So the other point I wanted to make is that Bruce Mate, those three designs that you see on that side of the screen, his own approach initially was that he was interested in staying just within the blubber layer. He was not interested in penetrating beyond these other tags. And for a long time, he stuck to those designs and he experimented with a number of them. Not all of them were used on animals. They were still pretty large. A lot of this was pretty much development, prototyping. But obviously the intent was to develop tags that would stay long time, and that in the order of 1.5 to 3 months. This is from the Montgomery report, ideally up to a year. Again, this was a pipe dream at the time. And so here are some of the recommendations about how to achieve that penetration. So a lot of those ideas were put forth back then, even though they were at that time hypothetical, really. So here's some of those examples again. This was the first one that was a satellite transmitting tag that Dave showed again this morning on a humpback whale in 1986. So after this achievement of the satellite transmitting tag, Bruce really just wanted to focus on how to achieve longer durations. And so this was the next evolution. A surface mounted tag, the pipe contains all the electronics, and the tines just penetrate the blubber and beyond the fascia. But there was a lot of drags with this type of tag. This was in the early 1990s. So for that reason, but Bruce made the final evolution was to package all the electronics inside a tube that would penetrate in the body, just like the original Watkins design, essentially much smaller. Of course, by this time, there was a lot of miniaturization in terms of batteries, in terms of electronics and so forth. So this was in late 1990s, 96, 97 is when we first started using these tags, which we still use to today. The form factor remains essentially the same. Obviously, our lab has not been the only one creating these kinds of tags. Alex has been doing this. Mats Peter Jorgensen, Nick Gales in Australia. So what you can see again is that convergence of tag designs that have been successful.
So one of the things that I think it's important to mention, and we'll cover this in detail tomorrow, is that all of these designs that we spoke about to now were produced for a number of reasons in separate parts. So the electronics package was produced by, different tech companies, and the attachment systems were produced by different scientists. And they all had what we call a transmitter anchor interface. Some of them had articulations and other features, but all of these tags had in common this one or multiple interfaces. And through follow-up studies, and I'm going to go to that soon. Well, just before I go to that, so this is how the anchor attached to the transmitter. So you can see here a threaded pin, and that threaded pin goes into that part of the anchor, and then there's the interface when you just screw the anchor on the pin. And we call these tags non-integrated tags because of the existence of interfaces. And through follow-up studies, and what is a follow-up study? Essentially, what we were doing is we were tagging whales, and we were trying to recite the whales to look at what happened to the tag after the whale is tagged, and what happens to the whale after. Into all the details tomorrow. Was one thing that was important here, and these are the many studies that we were supported by via NOP and through ONR that Dave mentioned today. We were able to document breakages at these various interfaces. So here's an example where the breakage occurs. So you can see that part of the anchor that screw on the tag, and you can see that the transmitter are lost, but the anchor stayed on. And what happens is that through what we discovered was that the loss of the tag, the loss of part of the tag, and keeping remains of the tag into the whale caused a number of potential health effects. Here's an example of one. Tomorrow, Juke and Marcy are going to cover the details. But clearly, that led us to obviously decide that we had to fix the problem. So here's the non-integrated tag. And how did we fix the problem? We worked with the tag manufacturer to create something we call an integrated tag. So we welded the transmitter to the anchor to make it more robust. And we started with that. And then we ended up creating what we call 3D printed. So the current tags, the most recent technology, are built as a single unit. They don't have interfaces, they don't have articulations for these designs, and therefore they're more robust. And we call that integrated tags. I'll cover this in detail tomorrow, but I'll come back to this later in the presentation. One of the things that we were asked to present is what kind of data we get from these tags. And these tags evolved over time in the late 1990s. Daniel mentioned the first tags that Bruce May put out. We had a limited number of sensors. The tags would provide primarily latitude and longitude, and the life expectancy of the tag was about a year or a little bit over a year. We didn't achieve a year, despite the battery is capable of doing that. But I'll show you that duration has improved. Then we have a second type of tag. There's archival. We have more sensors on it. We can collect temperature and depth, and obviously wet dry data to tell us, or to tell the tag we're dry or wet, send a transmission. We get location data, and we also get other information like depth and temperature. And these tags, typically, they're more battery hungry. Typically, they last about 90 to 120 days. And now there is a new system that Daniel's developing. It's called the RDW tag, which is very similar to this archival tag, but it also can obtain information on behavior and potentially foraging events. And we will cover that in a little bit as well, right?
Yeah.
So these are the main three types of tags that we have: Type C tags, and the different sensors that they have, and the different types of data they can provide. What are the pros of these tags? They're long-term data. They can last from weeks to months, and sometimes even years or over a year. They are embedded in the body of the animal. So unlike all of the tags that we've seen before, there's nothing left out for body contact. So they're less vulnerable to body contact, or less vulnerable to contact with the substrate. And there is proven robustness of the design, or at least some of the designs today. And the cons is, compared to, for example, the suction cup tags or the tags that John Kalamikita described, we get less sensors, fewer data streams. And because they are more invasive than all of the other types of tags that we've seen, they have the potential for higher risk of negative effects to the animals. So here's, I wanted to give you like a broad overview of the transmission duration. This is something that we're also asked to provide. How long do these tags work on the different species that we use? We call it a broad overview because the average duration of Type C tags are not very simple to calculate today. And the reasons are that many different designs were used by different research groups. We account for the different designs when we calculate averages. Some species have not been tagged with newer generation consolidated tags, and the North Atlantic right whale is one example of that. I think the last Type C tags used with the species was in the early 1990s. And the sample sizes for some of the species are very small. So despite these caveats, we put together a table showing what we expect in terms of durations for the many species that we work with. And you can see here that for many of them, we can get average durations of over 3 months, and in many cases, maximum durations that go beyond a year for many of the tags that we have deployed over the last 20 years. And this slide here is to actually show the evolution of tag duration over time, and we picked the right whales as an example. So here's the non-integrated tag, the tag that used to break with documented breakages. That tag had an average duration of about 47.6 days on right whales. And here I'm pulling all the right whale species together. And then the welded tags increased that duration significantly, and we started getting averages of about 112 days. And finally, the 3D printed tag, which is the design that we use today, and it's fully integrated, we're getting averages of about 211 days on right whales. So now we're gonna cover applications. How can we use this data? And we're gonna cover them under 5 different subtopics. migratory routes and destinations, discovery of new habitats, ecological findings, integration with other research methods, and management conservation applications. And I'm going to hand it to Daniel. Oh, okay.
So what we have here, let's see if I need to click it to start it maybe.
Yes.
Thank you. This is all our humpback whale tracking data together for the North Pacific, tagging done in the breeding areas, Hawaii and Revillagigedo Islands in Mexico, in the winter, of course, and their migration in spring and summer to various places in the Eastern and Western Pacific. Then tagging in the summer, fall months along the west coast of North America, as well as in the Bering Sea and Southeast Alaska, migration back to Hawaii, as well as to Mexico. In a couple cases, we had animals going to Central America. So that's just to give you an overview of the complexity of populations, breeding sites, feeding sites for humpback whales, and what we've been able to do. That animation contained about something in the area of about 150 tracks, spanning about 1995 to the most recent in 2019. So someone's career in there. Next.
So now we're going to talk about discovery of new habitats, and we wanted to show an example, which is kind of relevant to the discussions we had earlier. This is a satellite tracking study done in the South Pacific in New Caledonia. You can see New Caledonia here, the coast of Australia on the left of the screen in New Zealand. And tags were deployed on humpback whales in New Caledonia, and the animals migrated in many different directions. And they spend time in a number of seamounts around the South Pacific. And these was a discovery satellite tags. Seamounts are not areas where people are typically observing. It's water, there's no land around it. And it wasn't until the tags were deployed that we understood the importance of these features, these oceanographic features for this population.
Here's another case of an important discovery when Greg mentioned this morning the tagging of the western gray whales of Sakhalin. And the longest duration of those tracks were all animals that migrated to the Eastern Pacific all the way to Baja California, Mexico for the breeding season. And in one case, we had a return, Barbara returned back to Sakhalin. And in fact, her arrival was in May, which my understanding is earlier than all the other documented arrivals using other methods. So just to show some of the variation in migratory strategies along the route, we consider gray whales very coastal, but look at the basically high seas crossing once Barbara was returning to the feeding area through very deep waters, as well as, of course, shifting the paradigm between eastern and western gray whales and the complexity that's going on in that particular population. Still with gray whales, this is of the PCFG, the Pacific Coast Feeding Group. So these animals do not migrate to the Arctic in the summer. They stay in the Pacific Northwest coast, Northern California, Oregon, Washington, all the way up to Southeast Alaska. The animals were tagged off Central Oregon and Northern California. And what we see here is information about the timing of departure. So latitude 42 is about where the animals were tagged in late fall. And you can see that the timing of the migration is staggered. Not all the animals are migrating south at once, and not all of them are coming back at the same time. The other really cool thing that I find about this type of representation is that the slopes of those migrations are very constant, which is probably something that really needs to be investigated much more closely in terms of the optimal migratory speeds across animals. But other than that constant, really, there's basically a constant parade of animals between December and April. Some of them will start the migration very late. Some of them will start the migration very early. Some of them will continue after they arrive at the feeding areas the following year. They will continue moving up to Alaska. In a couple of cases, we had animals doing that. Next one. I also like the kind of data that tagging can reveal regarding residence time. In this case, in Hawaii, we had about 39 tags for which we were able to document the departure of the breeding site. So for those 39, the residence time in Hawaii from tagging to departure was on average 13 days, and a maximum of 43 days. But that also varied between sexes and age groups. So females with a calf, although we only had 3, click again, please, was a shorter duration of residence before they initiated migration, and getting increasingly longer as we have females without a calf and the males that tended to have the longer residence. Next one. A little bit about movements in the breeding area before they initiate migration.
So now we're going to cover a few of or a couple of examples of ecological findings that we had after deploying Type C tags in whales, and one of them relates to the critically endangered North Pacific right whale in the Bering Sea. Here, what we can see are 3 plots of the Bering Sea. In the first one on the left, that was during a what we call a warm year, and you can see that the individual moved around relatively to a relatively large extent. Compare that to the 2 plots, the one in the middle and the one on the right. Those are what we call cold years, and there is a tongue of cold water indicated in blue coming from the Bering Sea, and the animals tend to associate with that. So through this study, we're able to hypothesize that North Pacific right whales like the cold pool habitat. It's a good condition for reproduction of calamus, which is their primary prey there, and they move a little bit more around when the cold pool is not present in the Bering Sea. And that hypothesis was further tested and proved to be correct by acoustic data.
I'm going to use this slide to mention also our latest development in tag, and this is what we call the RDW tag. Alex already mentioned it a little bit. This tag has the same form factor of the other tags, but it incorporates an accelerometer, a pressure sensor, and the saltwater switch that can give us dive duration. But in addition to that, it incorporates several software development. We call it a smart tag because it has an adaptive algorithm. I don't have the time to go into the details of what that does, but there's a lot of software processing that goes on inside this tag, and the tag is able to detect events, feeding events, and then summarize them and transmit each feeding event per dive via satellite. So the tag is not recoverable, but we are able to document each of the dives. In this case, what we see is 6 humpback whales, 1 in purple and 5 in green. But the one in purple stands out more because each of those circles is telling you the number of lunch feeding events that occurred at that dive. So you can see a little bit of the spread of the movements of the animal, as well as the feeding behavior. That animal was farther offshore on the shelf break, diving deeper, which is what you see in this depth plots versus hour of the day, whereas the other 5 animals were on the shelf. We presumed that they were feeding mostly on schooling fish, whereas the deeper animal in purple that had many more lunch feeding events per dive was likely feeding on krill. So these tags basically allow us to document a lot of the variability on otherwise data that might look the same or that we might interpret as the same, a lot of variability between animals in terms of what they are feeding on, potentially feeding on. Earlier, there was mention about the ability to develop models, statistical models that predict habitat suitability, for example, and whether they are useful in the long term, especially in the face of climate change. And this just came out last week, where we used tagging data for, I think, 14 top predator species in the North Pacific Ocean, and including the blue whale, and models, predictive habitat models for that data throughout the 2000s. And then for these 14 species, we had data also collected during 4 marine heat waves that happened in 2014, 2015, 2019, and 2020. And during those heat wave events that were essentially unprecedented and that didn't have an analog, please click, please. We were using the data from the 2010s to validate and support the findings of the habitat model, to essentially find that the original habitat model that developed the data from the 2000s captured adequately the heat wave events. In this case, each heat wave event generated a different kind of displacement of the habitat, but nevertheless, it was predictable through those habitat models.
So I think earlier today, Greg mentioned about the importance of integrating the telemetry data with other research methods. And we have here an example of something that was very useful also for North Pacific right whales. This, the line in red is the track of a whale that was tagged by Paul Wade and collaborators in 2004. And while we were tracking these individual, there was a NOAA ship in the Bering Sea. And so we were able to direct the ship to the location where that whale was. And that ship, with the assistance of the telemetry data and acoustic data, was able to find the larger aggregation of North Pacific right whale detected since the 1960s. And during that time, they collected photo identification and biopsy sample from at least 17 individuals. And those led to the only estimation of abundance for these critically endangered species that we have today. So here's an example where you took the telemetry data and you use it to augment or integrate with other types of survey methods to improve significantly your knowledge of a critically endangered species.
In this case, we're using not just the tagging efforts, but we also collect a lot of photo ID in the field. And through photo ID, we can obtain the resite history of the animals, as well as anything related to wound healing, status of the tag, that kind of thing. So in this case, we're documenting additional connections that were not documented through tagging, but either resites or in different years. The other thing we do is with the biopsy, you also have the biopsy collection with the tagging. So in this particular case, the tracks are color coded by haplotype, and we're working out with Scott Baker and my master student, Liam Mueller-Brennan, on combining these 3 data streams, stable isotopes that we also derive from the skin, genetics, and resite information to have better inferences about foraging behavior and migratory destinations.
And finally, talk about how tag data, especially for Type C tags, are being used for management and conservation applications. We have a little animation here that shows.
Click on it.
Yeah. So this is a blue whale in blue, and this is the week of their lives in the northern Patagonian, Chilean Patagonia, and you can see the movements of the animals relative to ship traffic. This is not real time. It could be near real time, but it shows you the value of the technology to, for example, calculate risk assessment of ship collisions with this kind of whales on a relatively long-term scale. just in the interest of time, I'm gonna stop the animation and move on. Daniel?
Other uses that we have had for our tag data, this is the case of the blue whales. Peru started tagging blue whales in 1993. The last year we tagged was in 2017. And over that time, we were able to identify from the tag data, what you see on the far right of the screen is the overlap in home ranges, giving us an indication of the areas of highest use of blue whales off the west coast of the US in the summer feeding season. And what you see is that those two hotspots are exactly where the shipping lanes, the two main shipping lanes in California, or the ports are in the case of San Francisco and Los Angeles area. So through the identification of these persistent hotspots, we were able to generate information that could be used by a variety of stakeholders to better cite or modify the shipping lanes, for example.
And here quickly an example of the use of the track data from bowhead whales in the Arctic. I think this is the data set that Russ mentioned earlier today. These whales have been successfully tracked by Mats Peterheit Jorgensen, Laurie Quickenbush, and John Sita. And you can see that they were evaluating, where's the mouse? Oh, sorry. They were evaluating the overlap of migrating whales with areas of interest for the oil and gas industry in the Arctic.
In this case, there was a recent effort to revise the biologically important areas for cetaceans in US waters. And the most recent iteration of the BIAs, as we call them, use the tagging data stream as one of the data layers to inform the new BIAs for, in this case, for humpback whales in Hawaii. So what you see at the top is occupancy pattern derived from the tag data to objectively identify those hotspots and then outline those BIAs as one of the, as I said, one of the multiple data streams that went into the effort. And then the next one, I hope that John Kalamakinis will forgive me that I'll show this, just submitted to for the West Coast BIAs, where similarly we used the tag data, in this case for humpback whales, to inform and derive the new biologically important areas for humpbacks of the West Coast. So just examples of how we are taking the tag data in ways that require a bit of processing, a bit of modeling, a bit of development, quite derived products, but at the same time, they are the most useful for management applications.
And finally, just to mention briefly, and I'm going to cover this more tomorrow as well, is that we recently started working on a Type C tag that implants only in the blubber, and that's the subfascia model that Russ mentioned. And the idea here is that it would be a blubber tag for right whales. And we were motivated by the need to fill a gap between the limpet tags that were lasting approximately 10 days in average duration to the Type C tags that are currently not permitted to use with North Atlantic right whales. And so the planned features for this tag was to have the embedded electronics to take advantage of the electronics inside the body, minimize body contact and contact with the substrate, deployed in the blubber to minimize invasiveness, and be a medium-term tag duration that lasted on average 30 days. And also, if we develop this, and we did develop this tag could be a useful Type C tag for other smaller cetacean species like Rice's whale, Brute's whale, sei whales, and minke whales. And we're still working on the development. Currently, these tags on right whales are lasting an average of 23.2 days. And there are some improvements that can be made. And I'll cover this in more detail tomorrow when we talk about development of tags. And I think that's our last slide.
Our last slide is another one of those punctuated evolution moments where WWF, through their whale and dolphin program contacted a variety of tagging scientists around the world and achieved what had been impossible up to that time, which was for all of us to come together and contribute our data to form this global map of whale migration routes, which they call blue corridors is the name of the initiative. Oops, sorry, I guess that didn't transfer. So the Blue Corridors Initiative, the rationale or the justification behind that Blue Corridors Initiative was to time it with the UN COP meeting last year that was going to be dealing with areas beyond national jurisdictions, which is the high seas, the vast majority of the oceans, and was to be used as one more element of information and leverage to influence the development of jurisdiction that, or legislation, sorry, not jurisdiction, legislation that would help protect these areas that up until this point have very limited protection. So I love working with Alex. It has been an amazing working relationship, but to get, I think it was something in the order of 50 scientists around the world to get behind this effort and support it. It was that there. I don't know if we have time for questions or.
I think so. Yeah.
Thank you, guys.
Thank you.
So thank you guys very much. I think for questions here, we are going to move into a Q&A session here with Peter. I'm thinking because of the owl, we'll probably need our speakers coming forward. So it might be worth you guys coming up front. Bring your chair if you can. That way you can speak into the owl. And again, anybody in the audience, if you'd like to ask a question, we'll probably need you to come up. But this is the time we will take questions from the chat. And let's see who's driving, who's reviewing the chat. Lori? Okay, perfect. Okay.
I guess I'll come up here too. And
Lori,
Lori
they can
can,
hear you. You need a chair or you can.
I don't care. So you guys hear me okay in the audience?
So, yeah, I think
Okay.
our screen's frozen.
So we'll do Q's and A's. I mean, we're getting a few from the chat right now. One thing I wanted to clear up, there was one question about whether the talks would be available sometime after this meeting. And the hope is that we're recording the sessions and we'll make the recording of this day available, technology permitting. And also we'll check in on the different PowerPoint. We'll ask each presenter if they're comfortable to do that as well. So that's not going to happen immediately, but it will happen at some point. So I'll just go straight to the, look, they're coming in. Actually, I'm not going to be able to read. Okay. So I think the first question, if I read this right, is what requirement has there been, and this may go back to the previous questions from this morning, what requirement has there been from will also include concurrent studies on the short and long-term impacts from tagging, not just for permit reporting, but from the consequences of tag implementation, the impacts from chasing animals in vessels for up to several hours, for example, capture myopathy, or other stress or behavioral impact. So I'm not sure who wants to step forward and answer that. It's fairly long. Amy, do you want to give it a shot? Sorry?
Can
Oh,
you repeat the question? Sorry.
it was a long question. Okay. What requirement has there been from federal governments for tag deployment studies to also include concurrent studies on the short and long-term impacts from the tagging, i.e. not just for permit reporting, from both the consequences of tag implementation and the impacts from chasing the animal in vessels for up to several hours? And they include the questions of capture myopathy, stress hormones, and behavior impacts. Big question.
Right, so with regard to permit reporting, we do ask all of our permit holders to report the type of reactions that they're seeing, the percent of animals that have them, and then for tagging, as I mentioned before, the post-tagging monitoring, which I did see a question on there about health assessments as well. So we're looking for opportunistic monitoring. This sounds like this might be a little bit different from what we're asking for the permit reports versus does the federal government require studies? And to that, I don't have an answer.
Okay, Alex wanted to pipe up.
Yeah, I was just gonna mention that, you know, I think it's important that we do this kind of studies, but it's also important to note that behavioral responses are very difficult to evaluate because there's a number of stressors that are going on in your tagging. You have the approach, the boat, you have the tag deployment, often you have a biopsy sample, and these studies need to be designed to address those questions. And often asking someone who is allowed to tag 5 individuals is not going to answer the behavioral responses question, behavioral response question. So I think the government requires information that is useful, but we need to be careful about what we can address with the information that we get, and what we can address with properly designed studies.
And I guess I'd follow up just to say, this is obviously the kind of question we're asking in this workshop in the next couple of days. I really appreciate that it's on everybody's mind. How do you assess these impacts? And I think the sample size question was kind of interesting about how many you'd need to determine the impact. That's definitely something we want to discuss. But I'll let you have the final word, Daniel.
It was just that,
yeah.
It was just a clarification. I appreciate the questioner in regard to the hours of chase and capture or cardiac myopathy. And just to clarify that, at least from my experience, the tagging approaches that we do are not very prolonged because they generally don't lead to tagging success. If we're not able to approach the animal in the first few surfacings, it just becomes more and more challenging every time. So we usually abandon that particular animal and move on to a different animal within the first easily half hour, at the most 45 minutes, something like that. And then, yeah, so as far as the cardiac myopathy, things like that, as Alex was saying, trying to measure something of that nature along with stress or along with pain are the really difficult things that we are not able to do today.
Okay. Into the owl.
I'll just to follow up on that, I think part of the work we did with Western grey whales, and it's carried through to the guidelines, is that you do set your tagging protocols to try and avoid this kind of thing. We have limits to the amount of time you can spend with one animal, limits the number of approaches within a certain time. So there's a lot of safeguards built in, rather than putting a lot of effort into trying to understand the effects of them by designing an experiment. Maybe it's better to try and minimize them in the first place. And a lot of these things, as Alex said, are not dissimilar to other research techniques, getting photographs, biopsy samples. A lot of them have a common element there, and it's good to have those kind of practical guidelines.
That's great. The next question is actually on type A tags. For some of you who are sitting way in the back of the room, this is basically this question: have you experienced any damage to tags when they're deployed with too much force? And have you seen any differences in tag duration when deployed using crossbows versus pneumatic guns?
No.
The answer is from the back of the room has been no. You haven't seen tag breakage?
Early on in the evolution of the limpet tag, we did rarely have some tags break. And so in the period between like 2004 and 2010, we did have some tags. Usually what happened in that case was that the dart, one of the two darts would separate from the limpet tag. But we primarily addressed that and tried to be better about training people in how they deploy the tags. And so when the tags are following a path that's close perpendicular to the tissue that they're going to hit, then yeah, there's no difference between pneumatic and crossbow because we adjust the pressure of both of those. Or the ultimate velocity and net torque based on the weight of the projectile, the actual impact force on the whale. We measure that and we try to have an optimum range in which we deliver the tag on the whale. So the pneumatic devices that we use for limpet tags, for example, are equal to the devices that are crossbow based for the limpet tag. But for example, the ARTS pneumatic device has been used to deploy limpet tag device in Norwegian. And in that they measure that, they check that. And but in any case, it is still possible. I could purposely go out and break a limpet tag by doing it improperly, but it's pretty unusual now.
Okay, and I'll just let the people, I assume everyone heard that. If you aren't hearing it, put something in the chat, says speak up. I mean, the short answer was early on, they did see breakage. If you're using a crossbow or a pneumatic gun, you set the pressure so that they're pretty much equal. But if you're having trouble hearing, let us know. And Luke says he heard that. Next question. And this is a general question. Is there concern that bridging the blubber muscle interface will result in increased trauma associated with the tag due to the degree of slide that occurs between these two tissue layers? And I think this is one of the fundamental questions for discussion, but I'll see if anyone wants to tackle it at the moment. Other than that, I know it's a big question. I'm not sure about the answers. It's a question of research and concern. So
We have an expert on this topic here in the room, but I mean, obviously it's possible. I think, the way we try to minimize that when we're doing our work is to deploy the tags in places where some experiments have demonstrated that if shearing occurs, the impact to the animal is the minimal possible. So the tag is high on the body, close to the dorsal midline where the body flex is less, is what we try to do to minimize that problem.
Michael, do you want to pipe up here?
Yeah, Michael Moore. Alex and I did a study funded by ONR on this topic, and I agree exactly what he said. But I also want to add that there is always some slide and some trauma. And in the context of that, I do want to clarify, well, ask Russ for a clarification about your don't give a damn thing, because I think what you said, maybe I didn't hear it, that an animal doesn't give a damn whether the tag penetrates through the fascial sheath or not. Is that what you said?
That's not what I said.
Okay, what did you say?
What I said was the animal doesn't care if it's a type C tag or a type A tag.
Okay,
I presented those two classification schemes specifically for this, and obviously I did a bad job of it. The type A, type C, type A, type B, type C is the configuration of the tag. And, I could hold the tag over here, and I don't care if I'm tagging a right whale or a climbing dolphin. It's still either an A tag, a B tag, or a C tag.
Yeah.
Doesn't matter how big it is or how big the subject I'm tagging is.
okay,
Understand that?
I do.
What it does matter is where the tag ends up on the whale,
Fine.
which
Okay.
has to do with the size. So exactly the opposite of what.
We're
Apparently,
on the
right.
same page. I misunderstood you. Thank you.
Well, we clarified that. We got these two guys in the room talking to each other. Okay. I don't have more questions in the chat. I don't think, do I? Do we have one, Lori?
It was from earlier, so. It's Amy alluded to it already, but what types of post-tagging health assessment data are being required on permits and over what time period? What level of effort is being dedicated to obtain these data? For example, are dedicated surveys required, or can researchers just rely on opportunistic sightings?
So researchers can rely on opportunistic sightings. So we require researchers to collect images or high resolution video at the time of tagging to ID the whale and the location of the tag on the whale. But thereafter, our condition states researchers must make reasonable efforts to opportunistically monitor animals instrumented with invasive tags through tracking and resightings, for example, photographic, video, or genetics, to assess how the condition on the body is where the tag was, whether he's looking for breakage, tag wound reaction and healing, for example, severity of swelling, depressions, and coloration, animal health and behavior, whether or not a calf is present. So if a female was tagged, we're not talking about right whales here, and survival. We don't have a specific time period, but we're looking at, annual reporting. That's when that data is going to be provided, but we're hoping that that's going to be continued over time. But I think one of the things that I've been thinking about with regard to tagging North Atlantic right whales is they have to notify the permits office and the Marine Mammal Health and Stranding Response Program. I know HDR is really good about letting everybody know what tag was whale, what whale was tagged. Sorry about that. So the more people that are on the water that know what whale to look for, the better follow-up monitoring is going to be. So I think that's something that we could talk about, is how to, communicate that more, to get more people eyes on that whale.
Okay, we have a follow-up from Heather.
Yeah, correct me if I'm wrong, there isn't a requirement to submit data or photographs to the catalog from permits.
Correct.
So that, I know it's on the agenda for discussion, but that would allow, beyond that initial year, HDR is really great, but once that year ends, field teams change. A year from now, you may be looking at a golden sweep, and we would have the ability to prep teams in advance. Here are the whales to be on. We already do that for injuries.
Right.
We could add tag whales.
That way
So
we
that
can do long-term monitoring.
Correct.
So yeah, that's definitely something we can talk about as far as adding requirements.
Well, that's great. And just in case it was garbled, we're really talking about how do we use the information from the folks who tagged a whale, the photo ID information, get that cataloged and accessible to other researchers so that they can follow up, not just a month later or 2 months later, but over the years with individual whales. So I'll move on. Nope. Francis, sorry.
Sort of one comment to federal agency requirements to detect effects. Worst case scenario, an animal should die after tagging. If it washes up stranding, currently most of the necropsies of dead whales are done by volunteer organizations working in collaboration with the Marine Health and Stranding Program. And one of the problems is most whales washed up on a beach are on their backs. So it is often really difficult for a volunteer organization, first of all, to know if that whale was tagged or not. And then the effort to actually look for an impact is difficult because it's going to involve rolling the whale. Sounds very basic, but it's incredibly difficult and expensive to do, and it requires money. So I would say there's a plus and a minus. The stranding network is often challenged to be able to look for an impact. And when the Western grey whales were observed to be coming down the west coast of California, there was a fantastic effort to give all the stranding network photographs of the skin of those whales so that they could be, if they should strand, they could be necropsied, and there was funding available. Often that's not the case, and there's no funding available, and there's no way to know if that whale is tagged or not until you roll it. So that might be an area, if we talk about federal support, I think the stranding network, somehow having a better communication and support network for looking for those tags on whales that are lying on their backs. Like, if you know it's a need, you do it. If you don't think it's a need, you're not going to do it.
Okay, I'm going to move on to a more practical question. And I also looking at the people in the back, if you can't hear us yell, because I sometimes can't even hear the people right in front of me. But I'm going to go to another question first, which is how do you assess the appropriate length of the anchor with regard to the variation in species, age, and body condition? I think it's kind of a fundamental question for taggers. So I know people all around the room might want to answer, but who wants to just give the basics?
I would say because the anchors that Russell and John use are actually variable based on the species, that might be more relevant for them. Basically is up until the recent blubber tag, just one dimension, so there's no change. So I don't know if they want to add something about that consideration.
I'll just say for our tags, since our intent was to stay in the blubber layer and for our IFA, we scoured the literature for all the data on blubber thickness by species, and then made sure the dart length we were using was under those minimums. And that's why we had variable dart sizes.
Yeah.
Yeah.
So I'll try to repeat what John said, which is, with different species, they scoured the literature for blubber depth measurements and designed the tag to not exceed those depths. Is that a fair summary? Okay, yes, Kim.
I have a question on that. So this meeting is about North Atlantic right whales, not all whales, ostensibly. It's broader than that, but the focus is on North Atlantic right whales. And over time, North Atlantic right whales are smaller with less blubber thickness. So I was just curious, the data that you use for North Atlantic right whales, is that really current, so that you because I know the whale that died and stranded on Virginia Beach, they were really surprised at how thin the blubber layer was. So is that really current data, or is it older data that maybe not representative anymore?
And we don't have any right whales. Sorry, when someone wants to.
Well, there's a limpet tag, right whale. So I'm just wondering, based on what we know about blubber thickness, what's going on?
Wes, why don't you come forward so these guys can hear you?
So the part of the reason that when we started in the US Southeast tagging that we chose to use the off-the-shelf limpet tag was the maximum dart dimension that we had at that time. Well, actually we had tons of them, but the ones that you could standardly buy from wildlife computers were 7 centimeters of penetration. And so that's way below the average blubber thickness. However, there in fact are stranded North Atlantic right whales. Michael will probably pull it up in front of us and tell me how many are below 7 centimeters, but there are some that had, when they stranded, the vast majority of them were emaciated. And maybe you can comment on that, Michael, but based on the good data that they have from living whales, it's extremely unlikely that any of the whales that we tagged would have had blubber thickness less than 7 centimeters. But if it had been, we're talking about this much crossing the fascia. So we can get into the arguments later, perhaps tomorrow.
I'm not going to argue. I am going to say that I have very little faith in most necropsy right whale blubber thicknesses, and they were probably thicker than they were measured when they died. But also to say, Laura has been asking that we can talk more about the blubber thickness, blubber muscle interface today, because most of the people on the call won't be here tomorrow, and we've pretty much done some of that. But I just want to emphasize that it is a red line in terms of biophysics and biomechanics. That once you cross that, you're into a totally different organ structure, sensitivity. And so whether we talk about it today and or tomorrow, it's something that I personally think is a very important piece of discussion. And congratulations to Alex for going for that short tag. It's really cool.
And it's probably,
Sorry, I was just gonna comment.
Closer to the mic, Alex.
Comment a couple of things. I think it's important to talk about the blubber muscle interface because, obviously, it's a consideration. Sorry, that we're discussing here. But
Speak to everybody.
I
Speak
will.
to the people
I will.
and the people out there.
But I think with respect to the question that you're asked originally, Peter. So we have to consider that every whale has a different blubber thickness. And when you tag a whale, almost never we achieve full penetration because the tag almost always go in an angle. So you can only achieve full penetration if you have a vertical 90-degree deployment on the body of the individual. If it goes in an angle, penetration is shorter than the length of the tag. So for the work that I think Daniel and I are doing, we want to anchor the tag below the fascia because we know that that's what's going to give you the long retentions. I mean, I think there's a body of evidence out there saying if you want to get tracking these animals for 6, 7, 8 months, you have to go across that barrier. There's no technology today not to go across that barrier. So we make it a little longer because if the tag goes in an angle, then, you know, it's going to require a little bit more length to achieve that kind of a attachment. So it's really difficult to decide how long it is based on blubber thickness, but that's our rationale.
No, it's very clear. Thank you. That's great.
Just to illustrate it, I was gonna ask Sarah to pull up the illustration that we have of the body of the whale.
It'll just take a second.
The because what Michael was getting at is we can demonstrate it with this illustration also varies with the length of the whale. And it's never been measured in any mystici. And as you saw in the up about 5 or 6 slides. Oops, down. Keep going down. Fast and see a cross section of the body. Stop. So this cross section right here is taken from right here on a humpback because in the past we always tagged humpbacks right there. In fact, I still think even with Type C tags, that's the most.
So those in the audience, he's pointing to the aftermost cross section of the humpback whale, which you can see him pointing.
In the lumbar section. What?
Use the mouse.
It doesn't train.
Doesn't show up.
That's for us, but
Okay, well, but with right whales, southern right whales, Alex has been tagging actually cranial to the pectoral flipper insertion. And we don't know what happens up there is the truth, right?
In terms of what?
Slippage and what happens at the muscle blubber interface.
You say we don't know about the blubber thickness or?
No, we don't know what happens with slippage at this point in the body
In a right.
in a right whale.
Slippage, we don't.
So Michael Moore in the back of the room agreed that slippage we don't at the just post blowhole area. We aren't sure of the interaction or the fascial slippage, right? Francis has a point. Well, Michael comes up.
just a comment on this division between the blubber and the muscle. These are beautiful drawings, but in reality, it's not as if there's a clear black line at the base of the blubber and then a nice red line, with a hot red line at the surface of the skeletal muscle. Between the blubber and the muscle, we're talking about fascia. It's a sort of a fibrousy, loosey, some of it's tougher and some of it's accurate, and it has some mobility in that structure. I don't know about right whales, but in gray whales, at different times of the year, there is also fat that accumulates in that area. So between the muscle and the skeletal, between the blubber and the skeletal muscle, there can be abundant fat in whales harvested off Chukotka in Russia. You look at those animals, there is a spongy mass that's 2 to 3 centimeters thick of fat. So that's now another layer that tag may or may not embed it. Whereas at the end of the spring migration, there's nothing there. And so we have blubber depth, but there's some other tissue that also has
So
width.
I'll give you the right whale version. It's very, very similar. Right whales have 2 very substantial fibrous sheaths, one under the blubber and one above the muscle. And then quite often there are bottom layer to the top layer. And the width of that layer, I agree, changes significantly. But when it's got a lot of material in it, it's slimy. It's a very slimy oil. It's kind of like shampoo rather than butter or something like that. And so that interface that you're working with, you could actually probably happily use the Apple one.
So the upper one is,
Yeah, it's sometimes, you know, it's a 2 or 3 millimeter sheet. And then I've always wanted to map the whole thing because there's something weird going on in terms of how all these different sheets join together. I'm sure they're anatomically important. But, you know, basically, I think I totally agree that if you really want to stick around for a long time, you've got to get through both sheets and hook up underneath the bottom one. Then you've got 2 to work with. The question is the cost benefit of that. That's the question for this week.
agreed.
Okay, we have one comment from the chat about someone using Jeff Goodyear's capsule tags on right whales in 1988 and 1989. The durations of data collection were short because the antennas being broken off in surface active groups, not because of short attachment durations. It's a comment. And yes, we haven't actually talked about right whales and how they love to rub against each other. Very, very physically interactive. I don't know if that's affected your tagging success in southern right whales.
Sorry, can you repeat the question? That was.
Oh, no,
What Michael was.
I extrapolated from this comment that, you know, one of the things we've talked about with tagging in general is the behavior of whales, and right whales, as if I understand it, and I've actually studied them a little bit, are very physically interactive. So I was asking you, Alex, if with respect to the implantable tags, you've had problems with that, let's say versus limpet tags, for they can rub them off.
Part of what I'm covering tomorrow in terms of development is what kind of fixes we have to minimize these problems. And we used to have issues with not so much the antenna, but there used to be a portion of the tag that we call an epoxy bulb where the saltwater switch is. That part of the tag would break off. Didn't happen very often, but it happened. And that could have been caused by body contact or contact to the substrate. In the new tags, we eliminated that. It's a flat surface. The tags we deployed in Argentina and looking to Marcy, I don't think any of them that we were able to recycle had any problems with antenna or saltwater switch.
And actually, the rubbing as officially done in our favor because this is the tag end.
Oh, so Marcy's from the back of the room. It's actually done them a favor because it pushes the tag in
It's been
on
on
one or two occasions. Okay, we're getting toward the end of the time here, but I want to go to the room for the final questions. Randy Reeves.
Yeah, this is for Russ, so he's still up there. Have any North Atlantic right whales been tagged with Type A anchor tags other than in the Southeast US?
Yeah, and Veronique talked to that about the project that we're collaborating on in the Gulf of St. Lawrence.
Right.
There's a large quantity, 20.
Has none have been done in Cape Cod Bay, right?
No.
Okay, because I just wanted to throw this out there. It seems to me that given that you're getting to a point where you can expect some limpet tags to last for 6 days or 7 days on a right whale. Am I right about that? Is that stretched?
Yeah. So we had one that lasted 51 days.
Yeah, okay.
Well, but this definitely is very dependent upon the SAG behavior. In Gulf of St. Lawrence, when we tagged in the middle of SAGs, the tags came off instantly.
SAGs are surface active groups,
Right.
for those who don't know.
Yeah.
Yeah. And so Cape Cod could be a problem area for that.
Well, what I was thinking though was if you design the study such that you apply the tags at about the time when you expect the animals to be leaving Cape Cod Bay, you might have a chance of finding the route, some part of the route that they take to get into Gulf of St. Lawrence. That's what I was getting at. I thought about that for a long time, that would be an interesting.
Is Grace on the line?
So
I could speak to the whales don't tell us when they're going to leave.
Yeah.
They talk about leave, they come back, but Stormy doesn't know from the food. He knows when they're going to leave today. But you know, it's it's really unpredictable.
And I would comment back to that, that if you have a tag that can last 2 weeks, then you have a little float on that estimation of when they're gonna leave. So I think it is something we should talk about. It's a really good point. Lance?
I just wanted to point out that Dan Engelhop has deployed like 2 or 3 tags in the Mid-Atlantic around Virginia Beach. And it's sort of along this idea that, you know they're on the move. You have other assets there that can help find them. You kind of have an expectation that you're going to cover that path from off of Chesapeake Bay to maybe the Nantucket Shoals with that duration window. So again, it's that really targeted, like we want to know what they do in this really relatively small place. And it gets back to the earlier question of, refining your questions based on what's a reasonable expectation. Those animals are also solitary and moving, so they're not rubbing against one another, at least until they get where they're going. So yeah, I think it's just matching the question to the duration and the timing is useful.
Yeah, I have Alex, and then it's getting to like 2
I was gonna
minutes.
say
You
that.
better be thinking up your last questions.
That was precisely.
And Alex, come over here.
That was precisely one of the reasons that we wanted to develop this shorter blubber tag, because if the tag is embedded, then you eliminate for at least a large portion of time the problem with body contact and the sags and everything else, because the tag is not exposed. There's nothing exposed other than the antenna. And the oils can roll over the antenna and nothing's going to happen. So
Yeah, the development has been tremendous since, I work a lot with Jeff. They did great, but, things have changed dramatically. When Alex first started coming with us to test these tags, we literally had a hammer as big as the ceiling, smashing the crap out of them. So this area of the tag is now extremely well engineered, and it would take a lot to break the antenna. And if we go back up, you can just compare how vulnerable. Oh, sorry. Can you just go up 2 slides or 1 slide? This is on a different scale, but if you look here, the part of the limpet tag relative to how much anchor it has is tremendous versus how much of the Type C tag is exposed and therefore can be acted upon when whales bang into each other compared with its retention element. So just by design, because of the banging, this would definitely be a better design than that design.
Yeah, I'm looking for that last question.
Well, we
One
want
last-
to wrap up, I think,
Oh.
right? Yeah.
Well,
There
but
was a question asked really early today
Yeah.
that someone just flagged that hasn't been asked yet.
what is it?
At 12:23, Sarah Sharp asked a question.
I think that
We
was
did
the
follow up.
I think we've. Take a look. Did we answer it?
Yeah, that was the question Lori spoke up for.
Okay. We understand that we answered that question. That Lori read it out.
Sarah agrees.
Yeah. Okay. Sarah agrees. Good. Thanks, Sarah. Scott, I'm giving you the last question. You can change whatever you want. Just come up here and speak up.
Oh, I speak loud, I can speak. Right?
Yeah.
Yes,
Try it.
okay, so I want to talk about the C tags and the conservation application, those areas that were designated from the tagging data. It's terrific work. My question is, does it shed light on the sample size question that's been raised here? Because you had a lot of tag data for those critical areas to emerge. And clearly you waited for some period of time before you knew how much data you had so you could do it. And it also, I'm thinking in terms of not just duration, but actual satellite tag days that you're getting data back. So within the context of an area, you must have, 100 days or 1,000 days per square mile or whatever it is. So does that data share, does that provide any kind of insight into sample size that would be needed if you were going to try to apply tags to right whales to do management relevant, to provide management relevant information?
I'm gonna put Daniel on the spot first.
Sure.
He's the academic here.
I don't know what that means.
Here's the insights.
Go ahead, Dan.
I don't need to repeat the question, right? Was it heard?
I had Scott speak so loud, everybody heard the question. so
Everybody heard
Try
the
to
question.
paraphrase it. as
It's,
you
it's the question about sample size, which is a really, really good question. And if I can just go back to the beginning where the question is, can we even track a whale or which we are essentially systematically and methodically tagging dozen animals or so every year in the various study sites that we have. Yeah, it's a really important question because what we try to weigh is the logistical effort of the putting all the tags out and achieving a sufficiently large sample that we can make conclusions and that results that are reasonable, that are valid. And for the last 10 years or so, we've done a lot of the work with Navy funding from the Navy fleet of the West Coast in Hawaii area. And the goal of that work was monitoring. So we had to definitely be working with sample sizes that we would be able to say something conclusive and robust about that. And for what it's worth, I would say a sample size of 20 to 25 per year is what we feel was a number that we would feel comfortable generating results. Obviously, this is just very rule of thumb type of thing. It's much more involved. It's a very convoluted question about the strategy of how to deploy the tags in time and space.
But that's not what you use. That's not what you use for those analyses. So both for the humpback and the blue whale hotspot kind of analysis.
Yeah,
Those represented hundreds of tags.
that's cumulative for all of them. Yeah.
Right. So what were the numbers? I remember from the earlier slide that both your blue whale and your humpback tags were much longer duration than some of the other stuff. I think that's right.
Yeah.
So you had a lot of data, and how many tags are we talking about cumulatively for each of those two analyses?
Yeah.
That's beautiful work, but
So
it's a lot of tags.
yeah, I'll elaborate on that a little bit. So for example, for the Navy, we were generating annual layers of those hotspots based on the annual tag deployments. So that actually, thank you for the lead in, because that to be able to generate those hotspots or those home range overlaps, we feel that we need to have about 20 to 25 to generate something that is spatially cohesive, that can give you a good picture. For here, what I presented is the whole stack of 15, 20 years of data, because you can take it in however many, you can slice it and dice it in whatever way you want. So it's just for representation, but yeah, just reiterating that. But I will mention that, so humpbacks are the ones that we get the shortest duration, which is consistent with what John has found, everybody finds. For blue whales, fin whales, we get much longer durations. But yeah, overall, what I just said generally holds across all of them.
John Kalamakidis, last word. And loud.
It's a complicate the waters more, but I think it's less about the sample size of number of tags as it is about deploying them in representative and varied timing and areas of the population. It's different systems of looking at key habitats for the VIAs. We were comparing 3 different things, of which tags were one of them. We always have to factor in where were the tags deployed, what time of year. And if you cluster them all at one spot, what you worry is you'll get a biased result. And I would almost say it's less about the number of tags and maybe more about how you distribute them in a way that's representative of the population is the bigger challenge than just a numbers game.
So it's actually.
Okay, I'm gonna stop.
It's about-
I got the hook.
Yeah, we got.
I got the hook. Tomorrow. So we have
You
he hasn't got the hook.
He is the hook.
Okay. Much the sample size is way more complicated than seems to be coming out from the discussion. You can't say it's 20 to 25 because it depends on the question, depends on the distribution. It depends on the ancillary data that you have. It depends on what you're building it upon. Depends on changes through time. It's way more complicated than that. And there is no simple answer, but what it does mean is that you have to evaluate as you go on. And
It's an adaptive process.
it is.
Especially because the
So, I-
scientific answer to that question is different from the management answer to that
Right,
question.
we've got those questions.
Okay. All right, we've got those questions. Thanks, everybody.
Okay. So sorry for pulling the hook on that discussion. And I'm just gonna position myself so, yeah, that might be good for the. Yeah, sorry for pulling the hook on that discussion, but I'm just here to wrap up today's session. This morning, we started out with a few technical glitches that we overcame, and just now we ended off with a bit of a timekeeping challenge. So I'm just going to go over what we covered in between before we wrap up for today and prepare for the next day and a half. So we kicked off this morning with our colleague Greg Donovan. He set the stage for us through and amidst the technical difficulties. And so I'm just going to give a quick recap. He provided some food for thought when it comes to the purpose we are here today. He reminded us that with respect to the knowledge gaps that we are trying to fill, finding the solutions is really driven by the question and not the field technique used. And so he put forward that it's likely a combination of techniques that will best equip us to find solutions to the complex problems that we face today, or shall I say that the right whales face today. He also, bear with me, he also put up front and center one of the biggest and main topics or questions that we must ponder when faced with considering tagging as a solution. And that is the trade-off between the risks and the benefits. Following Greg, we had what I will refer to as the speed panel, a group of 6 experts with a range of opinions to help put into context what tagging could inform when it comes to right whales, as well as the concerns that must be recognized when contemplating these activities. We started with Véronique Lesage from Fisheries and Oceans, who spoke about the tagging project in the Gulf of St. Lawrence, the more recent summer foraging habitat for at least a portion of the right whale population, highlighting why the limpet tagging approach was chosen over other methods. That was followed by Colleen Coogan, who provided some context of protecting right whales in US waters where there are many competing factors at play, which are similar to those in Canada, such as important fisheries and busy shipping areas. She eloquently put forward, and I'll say it again here, that with a PBR of 0.7, we need to protect right whales anywhere where there is a risk larger than 0. Following Colleen, Clay George provided an overview of the right whale tagging work he was involved in the southeast US, reminding us that the whales travel a long way to arrive to the calving grounds, and our knowledge of how they get from point A to point B is influenced by our survey coverage. Sean Hayes put forth clearly and boldly some of the statistics that we are facing today when it comes to right whales, and stressed the importance that to adequately understand these animals, we should consider the value of tag data, and that we should do this with urgency, and that is before, as he put it, any whale that we tag or do not tag will be dead in 10 years if we don't act soon. Following Sean, Amy Knowlton provided a clear overview of the important concerns, primarily the effects of tagging on individual health and welfare, and that it is important that there is a clear study design and follow-up studies related to a question that can only be answered with tagging data before deciding to tag any of these critically endangered whales. And finally, Erin Mayer-Gutbrod provided us some perspective from the world of zooplankton and how important it is to gain a firm understanding on what right whales eat, where they are eating it, and how they find it. This, of course, was in the context of prey as a primary driver for right whale distribution. We then heard from Amy Sloan and Laurence Deneau-Tremblay on the legislative frameworks for tagging endangered cetaceans in both the US and Canada. And that was followed by a brief overview of the best practice guidelines, a culmination of a lot of hard work and the prominent best practice guide to inform cetacean tagging globally.
And in the room, a few points were raised with respect to health, and particularly have to consider that the right whale population has been shown to be experiencing significant health impacts, and how could this factor into decision-making? Between us and our lunch, we were enlightened by Dave Weller, who provided an overview of the history of right whale tagging, and who posited that gradualism that was later followed by punctuated equilibrium has led us to where we are today, a point in time when the science of tagging has evolved quite significantly with advancements that have been catalyzed over certain time periods. He left us with the idea that a cost-benefit analysis for tagging is needed, and that if such an analysis was easy to do, it would already be done. I want to take a moment, After lunch, we heard from the tag developers, Susan Parks, Alex Shorter, Russ Andrews, John Kalambokidis, Alex Zerbini, and Daniel Palacios. They provided information associated with different tag types and attachment duration. And while these researchers provided this information in nice, tight 20-ish minute presentations, 20 to 45, I want to say that I know there are hours and days and years of thinking and hard work that have gone into these developments. Seeing as we just recently wrapped up a discussion with these tag developers, I'm going to skip a summary of that particular portion, but I do want to take a moment here to sincerely thank all of today's speakers and panelists, and provide distinct recognition to these presenters who are here to openly discuss with us the work that has gone into tag development, the work that goes into permitting considerations, the work that goes into the work and preparation that goes into deployment, and how to consider follow-up evaluations. Now, looking ahead to the next 1.5 days in closed session, we will continue and engage in depth on discussions covering key information that could be gained from tagging based on the experiences of the experts in the room. There will be a particular focus on the Type C tags that we just reviewed this afternoon and their effects on large baleen whales. And we will discuss the utility of different tag types with respect to identified research questions. Last but not least, we will also consider data management and sharing, as well as the evolution of tag technology and design for the future. And so I'm going to wrap up quite quickly here, because I realize that we're about 15 minutes over time. But again, I want to thank each and one of the presenters, the panelists. Thank you very much for putting forward your thoughts and your ideas openly and transparently. We truly appreciate your participation. And with that, I would like to close today's session. I'd invite the steering committee if there's any last words. Very good. Thank you very much.
Just say a reminder for those who are in person.